Hi there, my name is Ian Reddin and I'm from the University of Kent in UK, and today I will be talking to you about our paper Drug-Adapted Cancer Cell Lines Reveal Drug-Induced Heterogeneity and Enable the Identification of Biomarker Candidates for the Acquired Resistance Setting. So a major problem with cancer treatment today is that many patients that initially respond well to therapies will ultimately stop responding to treatment. That is, they develop acquired resistance to treatment. This is opposed to intrinsic resistance, where there is no response to treatment from the outset. Here we have a simple diagram representing acquired resistance. The grey tumour cells are sensitive to therapy, but after treatment over time, changes occur in the tumour cells that allow them to adapt to the therapy, resulting in a tumour cell population with acquired resistance. Biomarkers for acquired resistance are required to predict treatment failure and alternative therapies, but this is difficult. Biomarkers are easier to identify for intrinsic resistance, as initial response to treatment is easy to measure. It either works or it doesn't. But the heterogeneity of cancer means that it is likely that processes resulting in acquired resistance are just as complex as the cancer itself. So preclinical model systems are required to determine biomarkers of acquired resistance, models that enable the repeated adaptation of a cancer cell population to the same treatment. Something that can't be done with clinical material, as patients can only be treated once. In this study, we investigate the use of drug-adapted cell lines as a preclinical model system. The drug investigated in the study is Y1155, also known as sepentronium bromide. It is an anti-cancer drug candidate that inhibits the BRCA5 gene, which encodes for the survivin protein that promotes cell survival. It does this via inhibition of caspase 9 and effector caspases 3 and 7, which prevents activation of apoptosis. Y1155 suppresses survivin levels, which then prevents inhibition of apoptotic pathways. A number of studies have investigated the potential of Y1155 against neuroblastoma, the most common extracranial solid childhood tumour. Treatment in these patients is currently poor, 50% relapse, and the 5-year survival rate is below 10%. The new drug development strategy is categorised survivin as a high priority drug target and Y1155 as a drug candidate for neuroblastoma. To investigate whether therapy-adapted cell lines can be used in preclinical model system of acquired resistance, we used Y1155 adapted UKF-MB3 neuroblastoma cell lines from our resistant cancer cell line collection. This cell line collection consists of over 1,000 cell lines that are resistant to a number of drugs. Here, a Y1155 naive UKF-MB3 cell line was cultured and treated with Y1155 until the cell line no longer responded to treatment. By doing this, we have 10 individual Y1155 adapted sublines that are characterised by the increase in Y1155 concentration compared to that of the parental, needed to decrease cell viability by 50% and 90%, as shown here. We also wanted to compare findings in the Y1155 adapted cell lines with Y1155 naive cell lines. This allowed us to determine whether an acquired resistance model could provide us with information that couldn't be gained from non-adapted cell lines with ovarian resistant status. It was possible to do this computationally using data from 2 studies that carried out pharmacogenomic screening on multiple cancer cell lines and multiple drugs. The genomics of drug sensitivity in cancer provided Y1155 sensitivity data for 915 cancer cell lines from 54 cancer types over 13 tissues, and this included 30 neuroblastoma cell lines. The Cancer Therapeutics Response Portal provided Y1155 data for 703 cell lines from approximately 60 cancer types over 23 tissues, and this included data for 12 neuroblastoma cell lines. And Y1155 sensitivity was measured as area under the curve. This is a figure that represents the area under a dose-response curve, and the higher the value, the less sensitive the cell line is to the drug. So TP53 has been linked with regulation of survivin. We looked at the TP53 status in the Y1155 adapted cell lines, and all 10 adapted cell lines in the study were wild-type TP53, so TP53 status is not associated with Y1155 sensitivity in these cell lines. We also compared the Y115 sensitivity between wild type and mutant TP53 cell lines in the GDSC and CTRP data, and we observed no significant difference in area under curve value between the two groups in either study. So it is unclear whether TP53 status is a reliable indicator of Y155 sensitivity. So cell lines in the GDSC and CTRP were classified by their survivin levels, either high or low. There is a small but significant difference in Y115 sensitivity between the two groups across all cell lines. The high survivin level group was associated with an increase in Y155 sensitivity in both GDSC and CTRP. There was no significant difference in Y155 sensitivity in the two groups in only neuroblastoma cell lines in either study. So, although we see an association between Y155 sensitivity and survivin level, sensitivity couldn't be predicted for an individual cell line based solely on survivin level. We still see many cell lines with a high survivin level that we would still class as resistant to survivin, and likewise cell lines with a low survivin level that would be classed as sensitive to Y155. In the adapted cell lines in the study, there was no consistent change in survivin levels compared to the parental cell line, as we can see in these Western blots. Survivin levels are similar across all cell lines compared to the parental. The difference in sensitivity to survivin depletion amongst the cell lines is indicated here by the cell viability compared to control. The higher the viability, the more cells are alive as less survivin has been depleted. The difference between these sublines indicates intra-tumour heterogeneity, that the sublines have developed different resistant mechanisms to Y155, and because of this, survivin level cannot be used as a biomarker of Y155 sensitivity in these adapted cell lines. The intratumour heterogeneity displayed was highlighted in the sensitivity profile of the Y155 adapted cell lines to another drug, vincristine. The IC50, that is the drug concentration at which 50% of the cells are no longer viable. So the IC50 of the cell lines vary dramatically between the cell lines. This heterogeneity in the sublines cultured from the same parental cell line really highlights in only 10 sublines how different mechanisms of resistance to a drug can occur in the same tumour type. Here we have Western blots that indicate ABCB1 and SLC35F2 levels in parental and adapted cell lines. ABCB1 is more commonly known as MDR1, or multi-drug resistant gene, and it mediates cellular efflux and is often responsible for decreased drug accumulation in drug-resistant cells. And SLC35F2 is a solute carrier that mediates cellular uptake. ABCB1 levels are increased in all 10 cell lines, adapted cell lines, compared to the parental. And SLC35F2 levels are generally lower in the adapted cell lines, but particularly in 4 of them. So this result would indicate that an increase in levels of ABCB1 and a decrease in levels of SLC35F2 have potential as biomarkers of acquired Y155 resistance. ABCB1 levels in the GDSC and CTRP cell lines confirmed an association with Y155 sensitivity. Cell lines in both studies were categorized as high or low ABCB1 level, and Y155 area under curve was compared between the two groups. The high ABCB1 levels were associated with a decrease in Y155 sensitivity. This was true across all cell lines in both studies, and particularly apparent in the neuroblastoma cell lines, again in both studies. Although the association between Y155 sensitivity and ABCB1 levels in the GDSC and CTRP data is clear, it isn't possible to predict Y155 sensitivity in an individual cell line based on ABCB1 level alone. So similarly with the SLC35F2 levels, we see an association with Y155 sensitivity in the two cell line studies. Again, cell lines in both studies were categorized as high or low. The low SLC35F2 levels are associated with a decrease in Y155 sensitivity across all cell lines in both the GDSC and CTRP, and across neuroblastoma cell lines in the GDSC And although we see the same pattern for the neuroblastoma cell lines of the CTRP, i.e. lower Y1155 sensitivity and low SLC35F2 cell lines, the difference between the groups was not significant. And as previously with ABCB1, although the association between SLC35F2 levels and Y1155 sensitivity is present, it is not possible to predict the sensitivity of an individual cell line based on SLC35F2 levels alone. So in conclusion, the difference in survivin depletion and vincristine sensitivity between the adapted cell lines highlights the heterogeneity of the sublines, and this heterogeneity suggests that the adapted cell lines have developed different resistance mechanisms to each other. And we demonstrate that cancer cell populations of limited intrinsic heterogeneity can develop various resistance mechanisms in response to treatment, and this is why it's essential to find biomarkers of acquired resistance to treatment that may not necessarily predict therapy outcome in therapy-naive cancer cells. A realistic way of doing this is by using preclinical models of acquired drug resistance, a method which we've shown here in this study. It allowed us to look at different acquired resistant mechanisms in the same cancer that wouldn't be possible in clinical specimens, as a patient can only be treated once. The emergence of ABCB1 and SLC35F2 as biomarker candidates for Y1155 resistance in our research highlights the potential of drug-adapted cancer cell line models to identify biomarkers that can be used in the design of individualized therapies. And I'd just like to acknowledge all contributors to the paper from the University of Kent here in the UK, and the University Hospital and Philipps University both in Germany. And the paper is available to read now on Cancers Online, so please do give it a read, and thank you for listening today.